Good day, and welcome to another class with Mr. Wilson. Today we're going to be looking at diffusion lab to be specific. And this is, of course, a CXC Biology with Mr. Wilson, Life and the Environment Explained. We know that you're home for COVID and you're wondering how we're going to be doing the labs. Now, many of the labs can be done virtually, and we're going to be showing you diffusion today. We're going to be looking at the effect that temperature has and on diffusion. All right, so let's jump right into the lesson. Now, diffusion, the first thing we want to know before we could dive into this lab, it's very important for us to understand what is this thing we're talking about, diffusion. Now, if we were supposed to go by textbook, diffusion is the movement of particles from a place of high concentration to a place of low concentration, and of course it takes place down a concentration gradient. Oh, I heard somebody saying blah 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 blah. What did he just say? All right, let us look at what we probably, or we could probably say it a little simpler. Now, diffusion is the movements of particles. Remember that matter is made up of smaller particles. This involves iron, and you will get to ion, not iron but ions, from a place where they are a lot to a place where there is less of that particle, and this takes place from high concentration to a place where there is low concentration, and that's a concentration gradient. But let us look at what we are really talking about. So here we have a glass with some water, and we'd love to have some syrup in this water. Right? Now, when we pour the syrup in this container, wherever the syrup is, that is pretty much high concentration. But over time, the syrup particles will continue to move through the water column until it's almost probably evenly distributed. Right? At that point, we can drink. So while it moves from that place where we have a lot of syrup to a place where there is less syrup, we are moving from a place of high concentration to a place of low concentration. I could give you another example. You're ready for school today, and you went upstairs and you pop your parents' cologne, the most expensive one. I don't know the names of these colognes, but you pop the most expensive cologne, and psst, psst, you got some onto your body. And your mom is downstairs in the kitchen, and she's, wow, I am smelling my cologne, and you're like, mommy won't smell it because she's not here, she's downstairs. However, mom is going to smell it because those particles are going to be moving from a place where they are at high concentration, that is upstairs where you just sprayed it, and they are going to be moving downstairs through the air to mom's nostril, and of course she's going to be smelling it, so you can't get away with it. You are busted, all because of diffusion. So diffusion pretty much is the movement of particles from a place of high concentration to a place of low concentration, and of course, like we said before, it takes place down a concentration gradient. What we're saying, it moves from high to low. Remember, at any time throughout this lesson, you can stop the frame, you can go back, you can play again however many times you so desire. However, please be reminded you need to be watching this with a friend. Please remember to share with a friend, get somebody to understand, to be a part of the story, so that we can have our big intellectual biology community. All right, let's move on. Now, diffusion. This fancy dancy word, diffusion takes place in solid, liquids, and gas. And you're going to be learning more about this if you're a chem student, or later in some lesson we will bring back this up, but we really don't want to get so deep into diffusion today. Now, diffusion is one of the processes that pretty much move substances through the body. It's just one of those processes. Now, when you talk about unicellular organisms, they would be using diffusion a whole lot. But we have a circulatory system, so multicellular organisms use some amount of diffusion. But of course, the circulatory system plays a big role. So diffusion is really, really important to us because it helps us to move substances through pretty much cell membrane. And we're going to be looking at that as we go further into the lesson. Now, diffusion is important for the process of gaseous exchange in organisms. And when we speak about gaseous exchange, I hate to talk about we breathe in oxygen and we breathe out carbon dioxide, because it's not necessarily true. I could get into the depth of that, but just follow my lessons, and as soon as we get there, respiration, I will be looking at that in details. Now, follow me. Oxygen getting into the cells and carbon dioxide leaving the cells. So when we breathe in and we breathe in air, we're particularly interested in the oxygen that is in the air. Remember that air is simply just a mixture of gases. So we are interested in the oxygen that comes in that air. Now that oxygen is going to diffuse into the cells because it's coming from a place where there's a lot. There's a lot. So we say that it is in high concentration and it's moving into the cell where there's just little oxygen. Why there's little oxygen in the cell? Because the oxygen that was in the cell, that oxygen was used for the process of respiration. Now, while that oxygen was used for the process of respiration, guess what happened? Yes, the process released carbon dioxide. And this carbon dioxide must also leave the organism. If it doesn't leave the organism, it could hurt the organism. So for that very reason, diffusion is important. So we are going to be having oxygen diffusing into the cell for the process of respiration, and we are going to be having carbon dioxide leaving the cell as a matter of excretion, right? In order to protect the organism and ensure that life continues. Now, it's a little different in plant. Of course, plants carry out respiration, but the plants would be using some of this in its whole day-to-day process. So let us look at the plant in a different frame. Now, the plant, of course, oxygen will diffuse through the stomata. The stomata are some tiny holes under the bottom of the leaf with some pores. The plant will allow oxygen to leave. And oxygen is pretty much a byproduct of photosynthesis. So look what happens. The oxygen will leave a plant through the stomata into the air, while carbon dioxide will diffuse through the very stomata into the leaf. Now, this carbon dioxide, why is it that the plant needs this carbon dioxide? Now, this carbon dioxide will be used in the process of photosynthesis to make glucose. So let's do that again. Animals will always use oxygen for the process of respiration, and they get to move this oxygen into the cells via diffusion. They will release carbon dioxide as a waste product of respiration, and it will leave the cells pretty much by diffusion. In plants, they are taking in carbon dioxide and giving off oxygen, and all this is taking place via the method of diffusion. All right, clear on that. Let's move on. Remember, you can pause, you can go back, but the most important thing, ensure that you understand. Now, we want to start the lab, and remember, there are some guidelines as to how labs are written. You probably want to pause this video, or you probably want to just hop on over to my lab rules. I have lab rules there for MM, for drawing, and for ORR, and they would give you in detail as to how the lab should be written. But we have just a simple draft here as to what a lab should look like, and we're trying to put together something here for both students and teachers. Now, this lab, remember the date must always be a part of the lab. This lab was done on October 9th, 2020. Now, the title, of course, is Diffusion. One might say movement, diffusion. Now, the aim, what is it that we intend to find out? We want to determine how temperature affects the rate of diffusion in liquid. And the liquid here is water. Now, the skill that will be tested, remember we said that you need to know the skill that you understand how to proceed with the lab. The skill that we're looking at today, we're looking at ORR, MM, AI. Now, be reminded, no more than 2 skills can be assessed for any particular lab. No more than 2 skills can be assessed for any particular lab. In this case, I have 3 there, and if you observe, I use one in a different color so that the teacher could determine where we're going with this lab. Are we going to be doing MM and AI, or are we going to be doing AI and ORR? The teacher has to make that determination. But remember, you can't assess more than 2 skills. Important to note for students and teachers as well, activity given must be completed in the time allotted for this session. So if it's a double session or a single session or 4 sessions, whatever activity the students will be given, it should be such that the students will be able to complete it in that session. And of course, it must be collected by the teacher at the end of a session. Now, if there's any lesson to learn, COVID is one to learn. You are there waiting and waiting and waiting and waiting and waiting, and 5 minutes later and 10 years later and that sort of a thing, and all the excuses come. Do not allow yourselves to be caught in that trap. Ensure that the lab is completed for the session. Ask your teacher, when will be our next lab? What will it be on? So that you can do some reading and prepare yourself for the particular lab. We're going to be putting up a whole lot of labs on my channel so as to assist both teachers and students. And at any time you see something that you wish to comment on, please just send us the comment. We are able to edit any of our content and to release it in a matter of days. Alright, so we are looking for this biology community to ensure that we're hitting perfection. Let's move forward. Now, after we would have had the aim, there would be the apparatus material. And we have spoken about apparatus and material several occasions. You can just hop back to one of our videos and you will see that. So what we use for this lab pretty much, or what will be used for this lab, is pretty much a beaker, measuring cylinder, spatula, thermometer, potassium permanganate, a refrigerator, microwave, stopwatch, petri dish. Remember, we always write the petri dish with a capital P because it's the name of a person. Alright, let's move right on. So after your apparatus, you're going to be having your procedure, or what we call method. Now, the teacher or the textbook might give you the procedure as is written here. But when you are writing it in your lab book for the lab, you must write it in past tense, and we did say for the labs that you are going to be writing it in past tense. So you want to observe here and ensure that you understand well. Now, this is given it in a bulleted format. Some are not really straight sentences. So you want to ensure that you can decide whether you're going to be writing it back in bulleted format for your procedure, or you're just going to write a continuous prose. Now, if you observe, there's an indentation from the left here. Remember, we spoke about that, so that you can write your scientific report here, your supervisor or your teacher, whomever it is. So you want to leave the space here, and if you observe, it's even down the line. Here we have in goal writing an instruction as to what is supposed to happen here. Let's move on down. Now, after your procedure, you would have had your diagram. And remember, if you're going to be using things from your diagram into the observation, then the diagram must be in front of your observation. All right, so you could draw your 3 beakers. You could say which one, label them A, B, C, which one has the hot water, which has the cold water, which has the room temperature water, that sort of a thing. If you're hearing some popping in my earphone, persons at work trying to reach to me, I do apologize for that, but I continue to make videos and I really can't break and turn off the system. I have to be doing both at the same time. I'm so sorry. Let's move right on. Results. Observation. What were our observations? Now, one of the observations keen to note is the temperature of the water. Yes, we need to know what was the color of the water before we started, because we really don't want to have any confirmational bias, right? We need to know what's the color of the water. We need to know what's the color of the potassium permanganate, so that when we report, we are reporting information that can be checked, information that is credible. All right, so you're going to be watching the video and you can make a determination as to what is what. Now the discussion, like we said, is which, where, why, when. In this case, you want to explain in your discussion why the results were the way they were. Why did the potassium permanganate not diffuse readily in this sample versus how it diffused in this sample versus how it diffused in that sample. You want to look at the comparison and give a good explanation as to why this happened. Remember, in your discussion you're going to have what we call background knowledge that pretty much starts the discussion, and it's pretty much an overview as to what diffusion is or whatever topic you're looking at. That's an overview of it, and then you're going to get into the explanation as to what is happening in the lab. And I did outline just now some of the things that you want to look at in your discussion. Why did the particle move the way they did? And why it did not move in this particular thing the way it did. How did the temperature actually impact or influence the movement of these particles? You need to explain that bit in your discussion. And then you're going to be upping and down to your conclusion. And remember, the conclusion is going to be pretty much linked to your observation, which is going to be answering your aim. So you want to look at, I think we wanted to look at the impact that temperature has on diffusion. So your conclusion is going to be saying here, temperature will cause the thing to move slower or cause the thing to move faster. Based on your observation, you are going to find that final position. Final position. So you must do the experiment in order to have an appreciation. Now, we're going to start the lab in a bit, but of course, you will not be able to use potassium permanganate at home. Now, I want you to listen carefully, and I want you to write these down. How you're going to do your own lab at home. Let's go. So I want you to get yourself a sachet, 3 sachets of Quenchade or Kool-Aid. Why we want 3 sachets? Because we want to ensure that the mass of the solute that has been used is pretty much the same. Now, if you don't have a measuring cup at home, take a teacup, fill it to the brim where you're sure that all the samples would have had the same water. Get yourself a soda pop bottle. If you are using whatever soda pop bottle you are using, please ensure that it's the same bottle you are using for all 3 samples. You are going to be pouring this water into each sample. That is, if you don't have a measuring cup to do the measuring. Of course, you are going to be putting 1/2 cup, one of your sample beaker or your makeshift beaker, which is of course your soda pop bottle. You're going to be putting one in your refrigerator for at least about 15 minutes, and you're going to be heating the water for another on your stove, on your microwave. But you want to ensure that you maintain that 200 or that cup full of water, so that all the beakers that you have made will have the same volume of water. Then you are going to be, you might not be able to use a thermometer because you might not have a thermometer. So you could just label your water hot, cold, room temperature. That would be good enough for your lab. You are going to ensure that you have your family participation, where you can add all 3 packs of Quenchade to the water after the 15 minutes it would have been taken from the fridge. So here you would have hot water, cold water, room temperature water, and then you are going to be adding your Kool-Aid or Quenchade to this volume of water, which is pretty much the same for all 3 containers. And then you are going to be starting your stopwatch. Now your stopwatch might be on your phone, it might be on your computer, it might be on the internet. Whichever one you are using, make sure that it is set to 0 so you can start immediately as you would have placed the Quenchade or Kool-Aid in these 3 samples of water. Then you are going to be observing the movement of the particle. You want to have your eyes locked on it, ensuring have the family with you. It's a family endeavor. Have the family with you, and you want to observe what is happening. And you're going to be recording the changes as they take place with the time on your timer. Right? So you probably want to document that in a table. You could also use a ruler to determine the distance, the Kool-Aid or the quenchade, the distance that this color is traveling through the water column, and how fast, at what time. And you want to look at the water, how purple it is, slightly purple, dark purple, or whatever color you are using, orange, red, green, blue, whatever color you are using. Just ensure that you are using one with color, not one that is what they say, lemon line or something like that. Don't use that one. Make sure the quenchade that you're using, it is color, and it would be best if you get all three same colors, and you'll be good to go. After about 10 minutes, you can end the experiment and make a decision as to what happened in your experiment, and you're going to write it up to follow the order that we presented here. Your teacher, of course, will provide some amount of guidance, and as the syllabus would have said, you are going to be doing this until you get it right. So we're going to be upping on into the actual lab that we did yesterday. It is important to note that we started the lab actually before we started recording, and I did that because primarily I was doing it alone. So I want you to get your family involved so that everybody can have an appreciation as to what is diffusion, the importance of diffusion, when probably is the best time to use an aerosol in your house to control whatever pest you want to control, or if you are using it for fragrance or that type of thing, or when you are cooking, how your neighbor will know what you are cooking, that type of thing. Everybody will have a good appreciation for diffusion as a part of the CXC syllabus. We're going to be hopping on now into the lab. Stay tuned.
So we see that specimen B is totally purple, and that happened at 1 minute 33 seconds. While we're still looking at the other two containers, the temperature is pretty close. Temperature here is 28 degrees Celsius, and over here the temperature is 22 degrees Celsius. We are 2 minutes and 38 seconds into the experiment. This is, of course, a diffusion experiment. So we are having a view from above. We're trying to look through the water column just to see how fast the rate of diffusion would have taken place in the hot water versus cold water versus room temperature water. So I'm sure based on what you're seeing, you could make a decision. I think you could clearly say how temperature impacts the rate of diffusion. Now, this is very, very important, because if you're going to be applying any pesticide, aerosol, then it pretty much dictates how fast it will move on a hot day versus how fast it will move on a cold day. All right, so this experiment is really, really important. Really important. It's a very important part of your CSEC syllabus to find out about diffusion and osmosis. And today we are working on diffusion. We have just used potassium permanganate with 200 ml of water. Temperature 22 degrees Celsius, 89 degrees Celsius, and of course 28 degrees Celsius. And like I have said, this is the COVID response to experiment. We're home, we're doing the experiment at home. I have some makeshift container here. They are just simply some little soda bottles. I actually use the scissors to cut the top off and to get the experiment going. But I did do some measurement with some measuring cylinder I brought from work. So I'm sure that the water, the volume of water is pretty constant across all. Same to the amount of potassium permanganates that we're using for the experiment. And of course, we're not using a stopwatch as would have been in the lab. I'm using the stopwatch on the computer, and you can see it. I'm just right here looking at it, monitoring the experiment. And from what you're seeing, I'm hoping that you'll be able to write up this lab, right? So we have used a beaker. We're using some modified beaker. We've used measuring cylinder. Of course, we use a balance to find the mass of the potassium permanganate. I'll soon tell you that. And we have used a spatula to actually get the potassium permanganate onto the paper. And of course, we use the microwave to heat up the water, and we had to use a refrigerator to lower the temperature of the water. And we just got specimen C from the pipe. And you remember it is at 28 degrees Celsius. Remember, you're going to be keeping your measurement in degrees Celsius, as we are going to be paying close attention to the SI unit. We're not going to be working with the imperial measurement. Remember, the syllabus requires you to work with the SI unit. And you would remember that for temperature, it is going to be Kelvin. So you want to hop on over to my video on measurement and manipulation. You want to look at that video we have there for temperature. The formula is right there, so you can convert degrees Celsius to Kelvin, because Kelvin is, of course, the accepted unit. Now, many laboratories in Jamaica, they are still with thermometers that might be degrees Celsius and might be Fahrenheit. But pretty soon we might make a change. It takes a little time for everything to change. But I think we're still doing well where labs are concerned in school. And I hope that students are really understanding the importance of using the SI unit versus the imperial measurement. We're still looking at the water column. We're 7 minutes into the experiment. And of course, you would have seen the 89 degrees Celsius. That water is pretty purple, like probably you would want to see a grape soda. Unlike the others, they look like they haven't been given much potassium permanganate. But of course, remember, we are looking at how temperature impacts the rate of diffusion. Now, if you're a chemistry student, you would remember that matter is pretty much in constant motion. And when matter becomes energized, you know that the movement is even greater. Now, if you're a physics student, you'd have an appreciation for that. You'd understand why the light wires have that sag. You'd understand why the train line has that space. You'd understand why the bridge on the road has the space, because the thermal expansion. So we have an appreciation pretty much for the work that temperature can do, how it can cause things to move very fast, very quickly. So we have to be very careful when we are working with the temperature, heat. It can really do some amount of damage, and it of course can do some amount of good. Even when we are working with things like liquid and gas, we made mention of some solid just now, but just look at what is happening in the container with the liquid and the potassium permanganate. There's a little movement here. You can see the potassium permanganate settled pretty much at the bottom of the container, and here you can see the potassium permanganate is all it has all diffused through the water column in this specimen with the hot water. And over here, the specimen from the top, wow, look at it, little or no movement. But we have to make a decision. So from what you're seeing on the video, after 9 minutes, this is what specimen C looks like. That's specimen B, and you can see the temperature there. And this is specimen A. I do hope that the lighting is pretty much the same. So just to look at specimen A and C, I'm going to be putting them beside each other, just to ensure that we have the same type of lighting and we can make a decision. So that's what's happening there. Just let me switch them around and see if that would be the same thing. Oh, so there's a lighting concern. So if you look over here, you have specimen A. Look at what's happening there. Look at it. Look to the bottom. Probably we'll have to get a ruler and probably measure the height. Let's move this out, and we're going to be looking at this. All right. So I'm going to be pausing the camera for now, and then we are going to come back and see if we can measure the height. All right. So there we have it. The left container is the one with the cold water. The one in the right container has room temperature water. So there you can make a judgment as to which of the container, what temperature diffusion would take place faster.